Good morning, everyone. Something's going on with my nose. I'm Steve.
And I'm Scott.
And together we're Backyard Musings, broadcasting live, Apple Valley, California.
Science and technology. That's what we do.
Yes, sir.
What we talk about.
In Brazil, researchers from the Federal University of Rio de Janeiro, UFRJ, along with collaborators from the University of São Paulo, have discovered a molecule called hevin, H-E-V-I-N, hevin, that can reverse cognitive impairment.
Nice name.
Let me say that again. Reverse cognitive impairment.
This is big.
The study, conducted in mice, revealed that this glycoprotein produced by brain cells known as astrocytes can increase the connections between neurons, the synapses, in both aged mice and in animal models of Alzheimer's disease. The research was published on 12th of February 2025 in Aging Cell. Would you like to know more? So it's reversing the...
That is wild. Quote, hevin is a well-known molecule involved in neural plasticity. It's naturally secreted by cells in the central nervous system that support the functioning of neurons and are known as astrocytes. We found that the overproduction of hevin is capable of reversing cognitive deficits in aged animals by improving the quality, of synapses in these rodents, says Flavia Alcantara Gomez, head of the Cellular Neurobiology Laboratory, the UFRJ Institute of Biomedical Sciences. So I can tell you right now, there's people I'm sure that would just be saying, sign me up for a clinical trial right now.
Clinical, yeah. The study was supported by the Ministry of Health, the Carlos Chagos Filho Foundation for Research Support, and the state of Rio de Janeiro, and FAPESP. It's important to note that there is still a long way to go before a molecule involved in reversing cognitive impairment can become a drug. This is a basic science study in mice. Another key consideration is whether the compound can cross the blood-brain barrier, which will require efforts to design molecules with this property and the same therapeutic potential. Well, we did a segment on that where there's it's argon. Wasn't it argon that could penetrate that barrier around the brain?
Yeah.
Wasn't it argon?
Pinpoint it, right? So let me just ask you this. If you had this disease, you were at the beginning stages of this disease, and you knew the outcome, right? The outcome is going to be not good.
Right, not good. Death.
Yeah. I would say, I want to try it. I want to be your guinea pig. I want to be your mouse. Because what do you have to lose?
You have nothing to lose other than your family will lose time with you if you die prematurely.
Oh, okay, but
nausea, vomiting, seeing stars in your eyes.
Yeah, diarrhea, the typical side effects of whatever drug we have nowadays. But I'm telling you, I'm sure people would say, sign me up, or sign my mom up, or my dad up, or whatever. This is really cool. Quote, of course, in the future, it will be possible to develop drugs that have the same effect as hevin. For now, however, the fundamental benefit of this work is a deeper understanding of the cellular and molecular mechanisms of Alzheimer's disease and the aging process. The originality lies in understanding the role of the astrocyte in this process. We've taken the focus away from neurons, shedding light on the role of astrocytes, which we've shown could also be a target for new treatment strategies for Alzheimer's disease and cognitive impairment, says Gomez.
By observing public data, the researchers found that hevin levels, that's spelled H-E-V-I-N, levels in the brain of Alzheimer's patients decrease compared to healthy individuals of the same age. With this information and using a recombinant viral vector, that sounds impressive, the group of the UFRJ Institute of Biomedical Sciences overexpressed hevin in astrocytes from aged animals and in transgenic animal models of Alzheimer's disease. So did they change them from male to female?
I was going to say, what is that transgenic? I don't know what that means.
I don't know.
In addition, the set of proteins produced by the brain cells, brain proteome of these animals was also analyzed. Comparing the animals with and without hevin over production, the researchers found that 89 proteins were differentially expressed. This stage of work was carried out in the Redux Proteomics Core multi-user laboratory of the Center for Redox Process in Biomedicine, Redoxima, okay. A FAP ESP Research, Innovation and Dissemination Center based at the University of São Paulo Institute of Chemistry.
Okay. Quote, the synapse depends on proteins to release a chemical signal from one neuron to another. Proteomic analysis showed that hevin, reinforced in astrocytes, regulates different groups of proteins involved in synapses. We observed an increase in synapses, or in other words, a closer connection between neurons and consequently better cognitive performance, explains Danilo Bilches Medinas, professor of the IQ-USP Department of Biochemistry.
In addition to identifying the potential of hevin to reverse cognitive deficits in rodents through behavioral testing, the researchers also observed that overexpression of the molecule in astrocytes did not affect the disposition of beta amyloid plaques in the hippocampus. Okay, what's a hippocampus?
It's part of your brain.
Oh, okay. A hallmark of Alzheimer's disease that has been the focus of disease studies and a target for drug development. Okay.
Quote, to our surprise, although the cognitive deficit was reversed in Alzheimer's model animals, there was no change in the content of the plaques. This highlights the complexity of the disease in terms of having a multifactorial mechanism. Oh, that would be crazy if the disease could, change according to the drug that's being given. Oh, that would be disastrous. This is illustrated by older people who have plaque formation but show no symptoms of the disease, says Felipe Cabral Miranda, a biomedical scientist at the UFRJ Institute of Biomedical Sciences and first author of the study.
Quote, although there's still no consensus among researchers, will work with the hypothesis that the formation of beta amyloid plaques isn't the cause of Alzheimer's. And the results of the study, by providing proof of concept for a molecule that can reverse cognitive decline without affecting beta amyloid plaques, support the hypothesis that these, although involved in the mechanisms of the pathology, aren't enough to cause Alzheimer's, he adds.
So it's promising. I mean, it's a work in progress.
I'm just grateful that people are working on it.
Yeah,
this is a good thing because
all across the world too. We've seen that. Oh, they're all working on it.
Yeah. So somebody, whoever gets the brass ring first, man, that's gonna be a big deal.
Billionaires.
Right?
Yeah.
Like,
I guess
what was it? Curie, right? Curie.
Curie.
Yeah, Curie. Now what the heck?
You mean the
Madame Curie, right?
Oh,
the one that she came up with something. I don't remember what her breakthrough was, but then we had the penicillin. That was a big breakthrough. Polio vaccine. That was a big breakthrough. I mean, this would be huge because this is just an awful disease.
Yeah,
it's terrible.
Yeah,
I know a lot of people that try to pinpoint it on diet and other things, but I don't know. But it definitely needs to be cured. That along with cancer.
Yeah.
Been working on both of those a long time. So.
All right, folks. Thanks for joining us. We'll talk to you again.
Yeah. We'll take care of us. Have a good day.